Hello friends, in this video we're going to see about biotechnology class 12. Okay, class 12, it is the second term portion, biotechnology. Under that, the requirements needed for a cloning vector. Here are the following requirements. Plasmids and bacteriophages have the ability to replicate within the bacterial cells. They have the ability to replicate within the bacterial cells. Plasmid, small circular DNA strand present in the cytoplasm of the bacterium. Okay, we also call extrachromosomal DNA. It can replicate independently. Okay, it can replicate independently. Autonomously, it will replicate. Okay, plasmid, extrachromosomal DNA, or small circular DNA strand present in the cytoplasm of a bacterium. Okay, that's plasmid definition. Bacteriophages. Bacteriophages is a type of virus that infects bacteria, or commonly you can call it as bacteria eater. Bacteria eater. Okay. So these plasmids and bacteriophages have the ability to replicate within the bacterial cells. And the second point, bacteriophages have very high copy numbers of their genome within the bacterial cells. Very high copy number of their genome within their bacterial cells. Genome, it is the set of chromosomes, okay, present in an organism. The genetic instructions, a set of genetic instructions present in an organism, that is called genome. So bacteriophages have high copy number of their genome in the bacterial cells. Third point, see if you are able to link an alien piece of DNA. Alien DNA means a foreign DNA or a desired DNA. Okay. So if you are able to link an alien piece of DNA with bacteriophage or plasmid, okay, we can really multiply its number. Okay. As many as copies we can make. We can multiply its number that can be equal to the copy number of the plasmid, right? Yes. Finally, vectors are engineered in such a way. Okay, they are engineered, they are manipulated in such a way. They help easy linking of foreign DNA and selection of recombinants from non-recombinants. So vectors are engineered in such a way. They help easy linking of foreign DNA and selection of recombinants from non-recombinants. So now I'll be telling you about the various features of cloning vector, right? So as I told you, the requirements of cloning vector, actually there are four requirements. Origin of replication, shortly called as ORI, okay? Origin of replication, selectable marker, cloning vectors, number 3 cloning vectors, number 4 vectors for cloning genes in plants and animals. I repeat, there are 4 requirements needed for a cloning vector. Origin of replication, shortly ORI, selectable marker, number 2, cloning vectors, number 3, and specially vectors for cloning genes in case of plants and animals. So here we are going to see only 2 features. Okay, we'll be dealing with origin of replication and selectable markers. The rest 2 will be continued in my next video. Okay, so first, origin of replication. So this is a sequence from where replication starts. Actually, replication starts here. A sequence from where replication starts. Any piece of DNA linked to this sequence can be made to replicate within the host cell. Okay, any piece of DNA. Okay, that's your alien DNA, foreign DNA, or desired DNA. When linked to this sequence, that is the replication sequence, origin of replication sequence, can be made to replicate within the host cells. So if one want to recover many copies of the target DNA, that is the desired DNA, it should be cloned into a vector. And of course, the vector will definitely produce high copy numbers. Okay, it has been engineered in such a way it will produce high copy numbers, right? So if one want to recover many copies of the target, it should be cloned in a vector. So that is about the origin of replication, shortly called as ORE. Shortly called as ORE. Okay, now we'll see the second requirement. Okay, we saw origin of replication. Now we'll see the second one, selectable marker. Okay, selectable marker. See, it helps in identifying and eliminating non-transformants and selectively permitting the growth of transformants. So this selectable marker will help in identifying and eliminating the non-transformants and recognizing selectively permitting the growth of transformants. Okay, two things. Helps in identification. Second one helps in elimination of non-transformants. At the same time, it selectively permits the growth of transformants. So we wonder what is the transformant? It's nothing but the procedure through which a piece of DNA is introduced into the host bacterium. Okay, that is transformed. Okay, a piece of DNA which is introduced into the host bacterium, that is called a transformant. Okay, so only the desired gene can be transferred to the host, right? The other thing may be unwanted. Okay, undesirable gene can be called as non-transformants. So which is necessary, which actually you want to multiply in high copy numbers, that alone we need. So that particular alien DNA will be introduced into the host bacterium. Example like genes encoding resistance to antibiotics. There are some genes that encodes for resistance to antibiotics. Such examples are ampicillin tetracycline. Okay, such as ampicillin tetracycline. They are considered as useful selectable markers for E. coli. Okay, especially the ampicillin and tetracycline are useful selectable markers for E. coli. Okay, so the remaining two, that is the third one, cloning vectors, and the fourth one about the cloning vectors, especially for plants and animals. We'll see that in the next video. Okay, hope you enjoyed this video. Kindly like, share, and subscribe. Thank you.